Hello everyone, welcome back to the week 3 question answer live session of the NPTEL course Fundamentals of Protein Chemistry. This course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur, and I am Bias Mukherjee, a research scholar from Dr. Shomode's lab in the Department of Bioscience and Biotechnology, IIT Kharagpur. And I am actually re-recording this session because in our Tuesday session where you all participated, there was an issue with the technical, um, there was a technical issue, and so the recording was not proper. And that is why I'm re-recording this. So in this course, we are going to talk about proteins. In the last session, we talked how amino acids come together to form proteins and how the secondary, tertiary and quaternary structure of proteins are made. We talked about contact maps in proteins and also other interesting topics. So in today's session, we will revisit the interactions between residues in a protein that enable the folding of a protein. Okay, and besides that, we will also talk about certain diseases that can happen because of mutations in proteins. Other than this, we are going to talk about the entire process of protein folding and look at different techniques that help to. Okay, we will look at crystallography and other techniques to study protein structure. Okay, so let's begin with today's session. Interactions in a protein. So in the last class, we learned that amino acids come together and get joined by peptide bonds to form a linear polypeptide chain of which again interacts with the residues in it to form the folded native state conformation, which is the biologically active structure of the proteins. Okay. So this is how the polypeptide chain looks like. It is just a long thread, and I was giving you the examples last class that the Maggi noodles that you see, when you boil it in water, it looks like this. It is having, it can just fold in any way, right? But then there are very specific hydrogen bonds and hydrophobic interactions, van der Waals interactions, disulfide bridges, and ionic bonds between oppositely charged side chains of amino acids, which are called salt bridges. And all of these various kinds of interactions help in proper folding of the protein. Okay, so if you want to look at the different kinds of interactions, here they are. There are hydrophobic interactions between non-polar amino acids, and there are hydrogen bonds, which are very important for making the secondary structures of proteins, that can be alpha helices and beta sheets. There are van der Waals interactions between the dipoles. Okay, they are weak forces and they are often long range forces. There are electrostatic interactions, as we saw in the previous figure. So these electrostatic interactions are between oppositely charged groups in the side chains of the residues, and they help in formation of salt bridges. Then comes in the disulfide linkages between cysteine residues in proteins, and these are very strong covalent interactions. For example, disulfide bonds are seen in insulin. Then comes in the pi-pi interactions. So pi-pi are stacking interactions between the aromatic side chains in proteins, and they help in contributing to the stability of the protein. Besides this, there can be pi-cation interactions, once again, between the cation and the aromatic side chains of amino acids. Right? So you can go through all of these different interactions to understand them in details. Now we will try to have a look at a few questions. So these questions are related to whatever we learned just now. Okay, so since you all are not there to answer the questions in the chat box, I will help you out with the answers. Okay, and I'll tell you how to solve these questions whenever you encounter them. Right. So which of the following residue pairs does not form a salt bridge in the protein? So there is glutamic acid and there is leucine. Now we just learned that a salt bridge is formed between oppositely charged residues in the side chains of the residues in a protein, right? So glutamic acid, you know, it's an acid. So the side chain is having another mm, carboxylic group, and your leucine, this is having what, aliphatic chain, right? So this is not exactly oppositely charged residues interacting. However, if you look at aspartic acid and lysine, aspartic acid once again is acidic, so it is negatively charged side chain is present. In case of lysine, a positively charged side chain is present. Now in case of aspartate and arginine, aspartic acid once again is negatively charged side chain and arginine has a positively charged side chain. Similarly, glutamic acid once again has negatively charged side chain and lysine has a positively charged side chain. And so therefore you can very well say from here that glutamic acid and leucine would be the right answer where the residue pairs do not form a salt bridge because they are not oppositely charged. For the next question, which of the following options contain all the information necessary to fold a polypeptide chain into its three-dimensional structure? Now, while teaching, I was giving you the example of your name, right? So, your name gives you your identity. It distinguishes you as a person. Similarly, the sequence of amino acids in a polypeptide chain gives it its identity and it tells it exactly how it should fold. Okay, so again, the sequence of amino acid, however, comes from the DNA sequence, and that is red. The codon is red, and then translation happens. But between the DNA sequence and the mRNA sequence that is transcribed from it and the actual protein sequence that gets formed after translation, there is a huge difference. Okay, so you try to answer. So there are a lot of untranslated regions. There can be different kind of rearrangements. For example, your VDG arrangement that you have studied in your immunoglobulins, right? So there can be so many different ways in which the genetic sequence is actually different from the polypeptide chain sequence. But here they are asking you what all information is necessary for a polypeptide chain to fold into the three-dimensional structure. So the first option is DNA sequence, second option is histone protein, third option is amino acid sequence, and the fourth option is RNA sequence. Okay, so here the right option would be amino acid sequence because once again DNA sequence is very much different from the actual sequence of the polypeptide chain, so it cannot direct it to fold into the 3D structure, right? Coming to histone proteins, histones just help in packaging the DNA and making it more compact. Okay, and it also assists in um, giving access to different kind of enzymes and transcription factors to bind to DNA and start the expression of a particular gene. But these do not participate in the folding of the protein. Histones are at the level of the DNA. Okay, do not come to the level of the proteins. Coming to the amino acid sequence, yes, amino acid sequence based on the uh, presence of different residues, there can be probability of different kinds of interactions of residues in its vicinity and also between residues that are far away from it. And so, yes, it is the amino acid sequence that drives a polypeptide chain to fold into a specific form. Okay, coming to the RNA sequence, no, that is not the correct answer. So, yes, the right answer in these two cases are option number A and option number C as we discussed. Okay. Now we move on to the next question. So this is a question on proteins. What type of interactions happen when a disulfide bond gets formed? Okay, so a disulfide bond is a covalent bond that gets formed between the two SH groups into different cysteine residues. Okay, so how do you think this bond gets formed? Is there reduction? Is there condensation? Is there oxidation? And is there glycosylation? So coming to reduction, the thiol groups, they are already reduced, so there cannot be reduction. Coming to condensation, no, it is not that. Oxidation and glycosylation. There is no question of glycosylation here. Okay. So let's look at the right answer. So disulfide bonds are formed between two free thiol groups and are sulfur-sulfur bonds. So the disulfide form is actually oxidized, whereas the free thiol is in the reduced state. So to form the disulfide, the thiol needs to undergo oxidation, and this is how disulfide linkage is produced. Okay. Now we will try to look at what happens if these proteins get mutated. So in our last lecture, we actually studied how important proteins are in our body. Right. Proteins are important as hormones, they are important as enzymes, they are important as the immunoglobulins and the defense mechanism in our body. They are important as transport proteins and also as your neurotransmitters in the brain. For example, GABA, right? So proteins are there all over in our body, and most of the bodily functions are carried out by proteins. A very nice way of saying this is that proteins are nano biological machines. Okay, so now what happens if there is some kind of mutation in the protein or at the DNA level? Say for example, I was telling you about these ultraviolet radiations in our class, that people, this is now commercialized, that you should always put on sunscreen and all that. And why is it so? Because there are all these harmful radiations, UVA, UVB radiations coming to us, and these have the potential to mutate our DNA, right? And so besides that, there can be so many radiations, there can be so many chemicals that actually mutate the DNA. And what happens if the DNA of a particular gene that codes for a functional protein gets mutated? For example, if you look at this, this is actually the gene for hemoglobin. Now you know that hemoglobin is one of the most important proteins in our body. It is the protein that carries oxygen to different places in the blood, right? So yes, what happens if the gene for hemoglobin gets mutated? So what happens here is there is a small point mutation. Point mutation means a single nucleotide gets altered. Okay, so what happens is the single nucleotide at the 6th position in this particular gene of hemoglobin, this gets mutated to a different base, right? Then what happens? 3 nucleotides actually make up your codon, right? And you know about the codons, right? Degeneracy of codons. So what happens? The first and the second nucleotide in the codon is very, very important, and you cannot go wrong with these two. But then for the third nucleotide, there can be wobble, base pairing, and so the third nucleotide, it can even surpass. If there is some kind of change in the third nucleotide, maybe it will not turn out to be a missense mutation, but it will become a nonsense mutation. But in this case, it is supposed to code for glutamic acid, but it becomes a missense mutation and it codes for valine. Correct? So what is a missense mutation? Missense mutation means that when a particular change in nucleotide results in the formation of a different amino acid in a protein. So a different tRNA recognizes this particular codon and it carries a different amino acid, which is valine, for the translation of this mutated hemoglobin protein. Now this is called sickle cell hemoglobin. Now you know about the quaternary structure of hemoglobin. We learnt about this in the last class. So there are 2 alpha chains and 2 beta chains in hemoglobin, right? But here what happens, the beta chain actually gets mutated and there is this valine instead of glutamic acid, and so this hemoglobin is different both structurally and functionally, right? So what happens is, because valine is a hydrophobic residue and glutamic acid was a hydrophilic residue, so the solubility of this sickle cell hemoglobin decreases. And what happens, at lower concentration of oxygen, more of these hemoglobin molecules come together and they stick to one another. Okay, because they are having this hydrophobic amino acid, and so they come together and they form an aggregate. This aggregate goes and blocks the blood vessel. Okay, and then what happens? It blocks the blood vessel and there is problem with delivering oxygen to tissues, right? Also, the shape of the hemoglobin changes. Now, this is the normal shape of your RBC, red blood cell. But then when sickle shaped hemoglobin is present, this RBC becomes like a moon. It becomes crescent shape. Okay. And what happens? This crescent shaped RBC, it is having very strange properties. It is having lower mobility. It gets degraded very easily. If it is getting degraded, then we are suffering from hemolysis or anemia, right? And another very problematic thing is that because the hemoglobin is now not very efficient in transporting oxygen to our tissues, there is very horrible chronic pain, and there is also organ failure due to insufficient delivery of oxygen. So severe pain, organ failure, chronic hemolysis, and blood vessel blockages. These are hallmarks of the disease sickle cell anemia. And sickle cell anemia is a very dangerous disease. This is also quite prevalent in India, and the patient requires huge amount of blood transfusions to survive. So this is very important that we have a knowledge of this disease and we try to come out with some kind of therapeutics or something to fight it. Okay. So let's try to answer a question on this. The question says, a point mutation in hemoglobin causes sickle cell anemia by changing a glutamate residue to a valine residue. Now, which type of protein structure is affected by the sickle cell anemia following this mutation? So, is it the primary structure? Is it the secondary structure? Is it the tertiary structure? Or is it all of these? Right. So, the first line in the question gives you the answer partially. Correct. So, in the primary structure, there is a significant change from glutamic acid, it becomes valine. So, yes, your primary structure is definitely going to get changed. What about the secondary structure? Yes, of course, the secondary structure also changed because we saw that this is a hydrophobic residue. And so, what is happening? It is undergoing more hydrophobic contacts rather than hydrophilic contacts, correct? So, of course, the secondary structure is also going to change. What about the tertiary structure? If you look at this, these actually come together and stick together and they form long rigid structures. So, that distort the shape of the RBC into sickle or crescent shape. And therefore, yes, even the tertiary structure is getting hampered. So, the right answer would be option number D, that is changing a charged amino acid to a hydrophobic amino acid will disturb all types of interactions in the protein. Hence, it will affect the primary, secondary and tertiary structure of the protein. Okay. Right. Now we come to the topic of protein folding. So this is a very interesting topic and I recommend you to go through the books that I have given in the reference, which are Voigt and Voigt, Biochemistry. There is also Leninger's Biochemistry. So Principles of Biochemistry. So please go through these two books. Besides that, there are so many different resources. There are different resources, websites and articles and reviews provided in the reference section of this PPT. Go through them. You can also watch very nice, interesting videos related to protein folding. These are going to give you a very nice idea of protein folding and the different kind of diseases that are associated with protein folding if it does not happen properly. Okay, so let's start with Levinthal's paradox. So Levinthal said that a single protein from this long polypeptide chain, it can fold into a specific structure, a specific 3D structure which is biologically functional, right? Now this single polypeptide chain, which is just a long thread, there are so many different combinations between different amino acids that can happen. Now, how is it that a single protein chooses exactly which structure is its native state structure, and it quickly transitions from this native unfolded primary structure to this well folded native state biologically active structure? Okay, to give you an example, consider that there is a residue, there is a protein made up of 60 residues. Okay, now every residue can maybe have 10 conformers or conformations, right? So what is a conformation? Conformation is actually the arrangement of the atoms of that particular residue in the 3D space. Okay, so how is it that the atoms are arranged? So what is the way the bonds are folded, and what is the rotation, and how is it that the amino acids, the different side chains of the amino acids are arranged, right? So that is the different conformations of a particular amino acid, different rotamers of a particular amino acid. So every amino acid, depending upon the number of bonds present in it, can have different number of rotamers. Okay, so let's say that there are 60 amino acids and every amino acid can have 10 conformations. So then the total number is 10 to the power 60, correct? Assuming that 10 to the power 11 seconds is the time per configuration, that is per conformation, the time is this. Then what happens is 10 to the power 49 seconds are needed to search all configurations. And that is these many years to actually come to the native folded state, right? But that is practically not the case. Practically proteins fold within seconds, even less than that probably, right? And there are so many different proteins in our body. If you take the examples of normal immunoglobulins, and there are so many huge proteins in the body, right? So how is it that those huge proteins are coming to the folded state so quickly? They might be having so many different conformations. So to answer that question, we have the protein folding model. Okay, so this is the protein folding funnel, and this funnel tells you that even though the rim of the funnel is huge, so rim of the funnel tells you exactly the number of possible conformations that a unfolded protein might have to go through to reach the native state. So native state is usually just one or two functional conformations that are very, very much active in nature, right? So how is it that from here, by exploring so much of conformational space, it goes into this uh, low energy, highly stable functional native state, right? So this is actually called the conformational ensemble. That is the huge number of conformations that are possible. So let's have a look at this. How do proteins choose the native conformation so quickly by not going through so many different possibles of intermediate transition states? How is it that they do it? Okay. So there are different explanations for this. Now if you look at this U, that is the unfolded state, this is how it looks like. Correct. From this unfolded state, there are different ways through which it can go to this native or folded state. So we look at them one by one. So the first one is nucleation. What is nucleation? Nucleation means that one tiny region in the protein, this first undergoes some kind of orderedness. Okay. Some kind of secondary structure gets forming at one small localized region, and then just the region next to that folds into some secondary structure. Again, the region next to that folds into some secondary structure, and this is how this actually propagates throughout the rest of the protein, and we get the native state structure. So nucleation means from a very tiny point, secondary structure formation begins and it propagates throughout the rest of the protein to give you the folded state. Okay, what is the second one? Second is secondary structure formation. So here what happens? Different secondary structures start forming at different regions of the polypeptide chain. After that, what happens? There is collision collapse. So in case of collision collapse, what happens? These regions having the different secondary structures, they collapse into one another to give you the functional tertiary structure. Okay. The other theory and the most accepted one, this is actually called hydrophobic collapse. So these two theories tell you that the most important forces are firstly the hydrogen bonds, that is from the primary structure to the folded state. The first and the most important force should be hydrogen bond. But then hydrophobic collapse tells you that the biggest priority should be given not to hydrogen bonds and other interactions, but to the hydrophobic interactions. Okay, so hydrophobic collapse tells you that all the residues in the protein which are hydrophobic in nature, they are pushed together into the internal region or the core of the protein, and all the hydrophilic residues, they are pushed to the outer region of the protein so that they can make contacts with the water and they can remain soluble. Correct? So this is all about hydrophobic collapse. Now we will have a look at the definitions of all of these different terms. Okay. Right. Let's have a look at the different terms. What is the unfolded state? Unfolded state was you in this picture. This is the unfolded state. Right? Okay. So, in the unfolded state, the polypeptide has no stable secondary or tertiary structure. It is highly flexible and has a random coil conformation. Okay. What about nucleation? So a small segment of the protein adopts a stable conformation which then acts as a nucleus, right? With one or more secondary structured elements for the folding of the rest of the protein. As I told you, propagation after nucleation, the folding process continues as the rest of the polypeptide chain adopts its correct conformation, right? This is all we talked about in the previous slide. So I have provided all this theory part so that the slides can be sufficient for you to be able to answer the questions in the NPTEL exam and also your future exams, right? So the slide should give you a very clear notion of whatever you're learning. That is why I've included the written part as well. So let's try to look at what is hydrophobic collapse. So the protein rapidly collapses into a compact form due to hydrophobic effect. In this state, the hydrophobic residues are buried inside the protein away from the aqueous environment, which drives the folding process. Now we will look at this in a very deeper way, okay? So we will look at the thermodynamics of protein folding, and this is what plays the biggest role there too. Okay, now coming to collision collapse, partially folded protein collapses into a more compact form due to hydrophobic interactions. And other forces such as van der Waals interactions, electrostatic interactions, biologically active or native state. This native state is the final and fully folded conformation of the protein where it is functional and stable, right? So in this state, the protein has a specific three-dimensional structure that is critical for its biological activity. I hope that is clear to you. Now we will look at this free energy tunnel. This is the protein folding tunnel. So what I told you is initially at the rim of the tunnel, you know that the protein is in an unfolded state. So from this conformation, it can go to this conformation, it can go to this conformation. You realize that all of these conformations, they do not have very defined ordered secondary and tertiary structures. So all of these are going to have extremely high entropy, right? So what is entropy? Entropy is the randomness or the disorderedness in a system, right? So you don't know in which way your noodle is going to fold when you are boiling it in water. You don't know which way it is going to go, right? It's just a long thread. But then when you know that if you look at your um, hand, okay, when you look at your fist, you know how compact and rigid and well folded it is. But when you look at your palm and all the fingers stretched away, then you can see that you do not know which way your fingers are going to go and which finger is going to go first. So I had explained this in the class, if you remember, but since we do not have the recording of that class, try to understand whatever I'm saying, okay? Now what happens is, as we move down towards the native state, this is actually the biological native state, and this point is called the global minima. Why is it called minima? Because this is the least amount of energy is there here. So free energy actually decreases as you go down, and this is the most stable conformation. Delta G is very much negative here. This is the molten globule. So what happens in case of molten globule? You see, this is a local minima. So there are many such local minimas in the folding funnel, and these local minima are points where the protein has been folded relatively compared to the unfolded state, but it is not the biologically active form. Okay, it is still going to undergo some kind of transitions, and there needs to be more amount of folding performed. So this is the molten globule, which is just a little less stable compared to the native state. This undergoes more folding to reach the native state, which is at the global minima, right? So the degree of nativeness increases as we go down, the free energy decreases as we go down, and the conformational entropy also decreases as we go down. Correct. So the free energy tunnel represents the decrease in free energy and entropy as an unfolded protein undergoes folding. So this is the unfolded state and this is the folded state. The number of conformational species, that is the number of different conformations that it can form, the conformational ensemble basically decreases and the semi-stable intermediates are formed. These are your different semi-stable intermediates. And ultimately, the highest stable functional native state is attained. Right? We already discussed this slide. Now we come to the thermodynamics of protein folding. So here they have explained this with a dimer formation, right? It is exactly the similar case when it comes to protein folding. So you can try to understand this, and the diagram is very easy for you to visualize the entire phenomenon. So if you remember the basic equation, which is delta G is equal to delta H minus T delta S. So in the class, I explained to you about this entire equation multiple times so that you don't have any issues with it, right? So let's try to recall the equation. It is delta G equals to delta H, which is your change in enthalpy, and minus T delta S, where delta S is the change in entropy, T is the temperature, absolute temperature, and delta G is your change in Gibbs free energy, a change in free energy, right? So what is free energy? Free energy is the energy that is present to perform some kind of work, right? And what is your enthalpy here? Here, because so many different bonds, all the hydrophobic interactions, hydrogen bonds, van der Waals interactions, salt bridges, all of these different kind of interactions are happening. New bonds are getting formed, bonds are breaking, bonds are being made, and that is why, because there are so many different interactions going on, a lot of energy is released. It is exothermic, and that is why delta G is actually negative. Sorry, delta H, that is your enthalpy, this is actually negative. Now coming to delta S. So initially when the protein was like this, you see that all these water molecules, they were interacting with the protein, and the water molecules seem to be very well ordered, right? Now what happens? The protein undergoes folding. So there is hydrophobic collapse. Now what happens? All the hydrophobic regions in the protein, these come together into one core of the protein, and from the core of the protein the water is actually pushed out. So the protein's entropy is decreasing. The protein is becoming more ordered and the randomness in the protein is decreasing. But for the water molecules, they were initially more ordered, and if you look at this, they are becoming very less ordered, right? So the randomness or the entropy for the water molecules is increasing. So the entropy of the system increases. That means your delta S, this becomes positive. So when the delta S value increases, then the entire term T delta S, that entire term increases in value. So what happens? From a negative term, if you subtract a more negative term, then again you get a negative term, and that negative term is your delta G, right? So delta H is negative, from that you subtract a higher negative term which is T delta S, and that is why you get a negative term which is delta G. So in this picture, if you see, two different um, polypeptide chains are coming and interacting with one another. It is similar. There is a kind of aggregation going on. All the hydrophobic regions are interacting with one another and they are packed together in the core of the dimer, and the water molecules are displaced and there is increased randomness. So hydration entropy is negative, hydration heat capacity is positive, right? Okay. So now we will go to the next part, which is how do proteins fold into the native state. Now the thing is that you understood why proteins fold and why they fold so quickly. Correct. They fold so quickly because they can undergo the hydrophobic collapse, and through the free energy tunnel, protein folding tunnel, you realize that this is how fast proteins can fold. Now, how do they fold? Okay, so the cell ensures that the proteins are always correctly folded and they are in their native state conformations. Because if the protein is not properly folded, the protein can undergo aggregation, there can be misfolding, there can be different kinds of aggregation, and there can be formation of these kind of variant species. So these are very dangerous. There can be amorphous aggregates, there can be oligomers, there can be amyloid fibrils, and all of this can actually lead to a wide range of diseases. All these different neurodegenerative diseases that we talk about, Alzheimer's, Parkinson's, Huntington's disease, these are not very rare, and in everyday life you come across people who are having these neurodegenerative diseases, especially people in their old age, and it is very difficult to treat those patients, very difficult for medicines to work, and most of these diseases are because of misfolding of proteins. Okay, so we will try to see how the cell prevents this. Okay, so here what we see in the figure is that nascent polypeptide chain is getting produced by translation, and the nascent polypeptide chain is actually unfolded, unmodified by post-translational modifications, and that is why it is called nascent or very virgin polypeptide chain, right? So then it undergoes folding into an intermediate form. So from this intermediate form, there are different helper proteins which are called the chaperones and chaperonins. So these proteins help in folding this intermediate into the native state, right, into the native functional protein. Then there are different checkpoints in the cell. So these checkpoints ensure that the protein that is going out of the endoplasmic reticulum, it is truly well folded. If it is not well folded, it is sent back into the ER and it is made to fold once again. So the chaperone and chaperonin gives this protein multiple chances to fold into the right structure. Even after that, if it is not folding into the correct structure, if there is kind of misfolding, then what happens? The protein is marked with ubiquitin for degradation. So multiple ubiquitin molecules are added to the protein molecule, and then through the proteasomal degradation pathway, this protein is sent into the proteasome, into the lysosome, where what happens? There are all these digestive enzymes, proteolytic enzymes, which actually degrade the protein. It chops up the protein into peptide fragments, right? And this is done to prevent the formation of all of these unusual species inside the cell, which anyway cause the cell to undergo autophagy. So the cell kills itself to make sure that all of this is gone. So your body cell is getting lost in the process, right? This is how the body ensures that the proteins are folded correctly, and it prevents the formation of different kinds of misfolded species and diseases associated with them, right? So protein folding is mediated by chaperones. Chaperones are mostly heat shock proteins. So heat shock proteins are proteins that express more when there is some kind of stress condition. Now the stress condition here is what? All these misfolded proteins, right? So heat shock proteins and these basically the stress proteins. Stress proteins can express themselves when there is some kind of different temperature, different pH conditions. There is some kind of chemical, and in such, and there is less oxygen. So in these kind of different stress conditions, these proteins express. So the most commonly known proteins are HSP70, HSP90. Now 70 and 90 are mostly the Swedberg unit. Have a look and check if that is the case. Okay, I might not be saying the right thing, so have a look at that. And then there are chaperonins. So chaperonins are also helper proteins. There can be GRO-ES and GRO-EL in E. coli. Okay, so we will look at the mechanism of these chaperones and chaperonins in the next slide. Now let's look at what happens to proteins if they are misfolded. Okay, so unfolded and misfolded proteins cannot pass the ER-Golgi quality check and are degraded into constituent amino acids by proteasome mediated degradation, the one I told you about. And misfolded proteins can cause different kind of disease conditions like cystic fibrosis and accumulated amyloid fibrils and insoluble aggregates, several neurodegenerative disorders like Alzheimer's, Huntington's disease, and so on. Right. Now we will have a look at how these GRO-ES and GRO-EL works. So if you look at this, I was telling you as an example in the previous class that this GRO-EL is kind of a container. Okay, it's like of a jar. And this GRO-ES is like a lid. You close the jar and you put the lid and you put this unfolded protein inside. Then you take ATP, and ATP means a lot of energy. So with a lot of energy, you churn and shake the container, and then what comes out is the folded protein, right? This is how this particular thing works. Go through the detailed mechanism. I have provided you with so many different links with the book references. Please go through them. This is just for an example that I'm telling you to remember this. Like you can remember this in this way, but look at the actual mechanism to understand exactly what happens in this case. Right. Now we come to the chaperone mediated folding. So this was all about chaperonins. And what comes in for the chaperones is that protein is getting translated, polypeptide chain undergoes folding into intermediate structure. If there is misfolding, then what happens? The chaperone mediated processes make sure that they are folded into the native state structure. So if that does not happen and there is still misfolding, then what happens? This protein gets labeled with multiple ubiquitin. So there is polyubiquitinylation, and this protein is targeted to go to the proteasome where it undergoes degradation into fragments of peptides by proteasomal enzymes. Right? If the misfolded protein is not checked properly by the quality check system of the cell, then it will undergo aggregation and it will result in the formation of different kind of diseases. For example, Alzheimer's disease. Now, what kind of misfolded aggregate of protein is responsible for what kind of disease? That you must remember. That is a very important question when it comes to your exams, right? So you'll get match the following for this. Please try to remember exactly what kind of misfolded protein species is found in what kind of disease. Okay. So for Alzheimer's disease, it is tau. For Huntington's disease, it is huntingtin. For Parkinson's, it is alpha-synuclein. For your prion disease, it is this. For Alzheimer's, it is amyloid beta. So please remember all of these. Now, HSP70 binds to the hydrophobic regions of the nascent protein or the misfolded protein and helps them to fold correctly. This is what is the function of HSP70, which is a chaperone. HSP90 assisted in the maturation of the specific protein involved in the signaling and other cellular processes. Okay, I hope that is clear. Now we will look at the bigger picture once again. So under physiological conditions, the polypeptide is expected to be in a soluble form. It is expected to assist in keeping the tissues intact and forming intact tissues, and healthy neuronal function is supposed to be going on. But what happens when there is internal or external stress? Now stress can be varied temperature, varied pH, introduction of chemicals. Stress can come from even the tiniest of circumstances such as lack of proper sleep, right? And consumption of food that is not healthy. Plus there can be DNA mutation, which can be because of exposure to radiation, exposure to chemicals, exposure to different kinds of radioactive elements. There can be DNA mutation, there can be protein overproduction because of altered expression of gene, there can be cancer, there can be tumorigenesis, and there can be immunogenic insults, that is bacterial and viral infections. There can be also allergens, right? Allergic reactions. So these are the different kinds of stress because of which proteins can get misfolded. Then what happens if the protein is misfolded? So there is ubiquitinylation and proteasomal degradation, right? And then the tissue and everything else is restored. Normal functioning of the body is maintained. There can also be autophagy of the cell to restore all of this normal functioning. What if the protein clearance system is damaged? What if it is not happening properly? It can actually lead to cancer. It can progress into neurodegenerative diseases. There can be abnormal inflammation and immunological issues can also be seen. Protein aggregates can be there, and there can be genetic and pharmacological manipulation of the proteolytic systems need to be performed so that this can be restored. Intact protein clearance system can be restored once again. Okay. Now we come to the process of denaturation and unfolding. So this is a very basic, very nice, interesting thing. What is exactly denaturation? What is exactly unfolding? So unfolding is actually a reversible process. The proteins, secondary, tertiary, quaternary structure, these can be broken when there is unfavorable temperature, unfavorable pH conditions, and unfavorable buffer system. Right? The protein can undergo unfolding. But it can always be restored when there are again optimal conditions. What about unfolding? What about denaturation? So denaturation is not a reversible process. It is an irreversible process. So one of the examples is that when you run an SDS page, you want to visualize proteins as monomers. Then what do? You actually heat it to nearly 100 degrees centigrade. So because of this heat, the protein is irreparably damaged. The primary structure only sustains because it is made of covalent linkages. Baki everything else is gone. So that is denaturation of the protein. Denaturation can be caused by very strong chemicals, heat, bases, acids, and denaturation agents. Okay. So see, denaturation means that there is loss in the three-dimensional structure, abrupt unfolding over a narrow range of temperature, and this is how it happens. Percentage of maximum signal, that is the fluorescence that you will get, and this is temperature, and this is how the denaturation or unfolding of the protein is seen, right? Now what happens is denaturing agents. So when you're giving denaturing agents such as heat actually affects the hydrogen bonds, pH, this affects the electrostatic repulsion, disruption of hydrogen bonds, okay, and alteration of the different pKa and the pi of the protein, right? There can be organic solvents such as alcohol, acetone, urea, guanidine, hydrochloride, detergents, etc., which disrupt the hydrophobic interactions in the protein. So this is actually guanidinium hydrochloride. As you increase the guanidine hydrochloride concentration, the percentage of unfolded protein also increases, right? This is a sigmoidal curve. Now what happens is, what if you want to study the structure of proteins? So this is the conventional experimental way in which you can study the structure of proteins. There are different techniques. For example, nuclear magnetic resonance spectroscopy is one of them. Our lab performs NMR spectroscopy, and this is useful for small proteins, okay, less than 20 kDa. But what if the protein is huge? Let's say it's a 60 kDa protein. For those, you can go for protein crystallography. So protein crystallography actually fixes the different atoms and the different entities of the protein into very defined arrangement, and then a crystal is prepared. So how do you make a crystal? The protein needs to be subjected to definite salt, precipitant, pH and buffer conditions. The protein concentration, quality, stability and purity must be appropriate, and then the physical variables such as temperature, setup, ratio, drop size, all of these need to be manipulated, and if all of these are ideal, all of these are perfect, only then you get a crystal. Now it is very difficult to get protein crystals. Also, some proteins do not give you crystal at all. It depends upon the amount of orderedness. It depends completely on the sequence of the protein. Okay, if the protein is very disordered, then it is very difficult to get crystals for those proteins. If you look at the protein data bank, which is available online, if you search by typing PDB, and if you look at the structures of different proteins, then you will see that the proteins that are disordered. Okay, they will maybe have only the middle part with some very ordered folded region or domain crystallized, but then the N terminal and the C terminal portions are missing, because N and C terminal are very dynamic and it is very difficult to crystallize dynamic regions in a protein, okay, that are disordered. So you can always look up the protein data bank and have a look at different proteins there. So now this is how a protein crystal looks under the microscope. So you take the crystal and you subject it to X-ray. Now X-ray exposure can be through home source or it can be in a synchrotron. Find out in India where we have this synchrotron. Okay, it is just there in one or two places. Find out exactly where the synchrotron is. So a synchrotron is a huge setup where X-rays are going around in circles. And these X-ray electrons are going around in circles, and from here the X-ray is emitted and this X-ray is subjected to fall on the crystal. Then what happens, based on the position of electrons in the crystal, you get a diffraction pattern. So from this diffraction pattern, this is actually called X-ray diffraction, and that is why the entire process is called crystallography and X-ray diffraction, right? So you get this diffraction pattern. From this diffraction pattern, you actually analyze it and you come to get the electron density map. So this tells you about the different residues present. So based on the resolution, it is easier to identify the residues. If the resolution is very good, then you get to know exactly what is the structure. So then what kind of residue should it be? If the resolution is poor, it is all the more difficult, right? And then there is fitting and getting the phase correct. Then there is structure determination and refinement to get the 3D model. And it is an iterative process and you do it over and over again to ensure that everything is correct. The model that you're getting is just right. Okay, so let us begin with the assignment. We already solved a few questions at the beginning of today's session. So we'll continue with the others. Okay, so this is a graph telling you about the fraction of unfolded protein that is formed after the protein is heated to a temperature of 100 degrees centigrade starting from 10 degrees centigrade. Okay, so as you heat the protein, because of heat, you know that the hydrogen bonds and other interactions in the protein, they are destroyed, they are broken, and so the protein keeps on unfolding. As the protein unfolds, there is some dye that is added and it goes and binds to the interior of the protein. It goes and binds to the hydrophobic regions in the protein and some more of fluorescence is detected. So this is the fraction of the unbound protein. So what is the melting temperature of the protein? That is what they are asking, right? So melting temperature is the temperature at which 50% of the protein has been unfolded. So from this figure you can say that at what? 0.5, that is 50% of the protein has been unfolded. So it is happening at a temperature of 45 degrees centigrade, right? So the temperature at which the fraction of the folded and unfolded proteins is equal is known as the melting temperature, and it is 45 degrees centigrade. Correct? Now we go to the question on protein crystallography. So what is obtained after the X-ray diffraction of proteins? Is it molecular density map? Is it nuclear density map? Is it electron density map? Is it atom density map? Right? We discussed about this just now, so you know which is the right answer. The next question is, which of the following amino acids sequences is most likely to be a part of the core of the protein? Now amongst these combinations, you have to find out exactly which combination has the most number of hydrophobic residues, because they are more likely to be at the core of the protein. Correct? So The right answer would be C, electron density map for the top question, and for the one after that it is this option, option number B, where phenylalanine, histidine, methionine, cysteine and valine, they are all non-polar hydrophobic side chain containing residues, and therefore they have the most likelihood to cluster in the interior of the protein. Correct? Now try to answer this question. The molten globule state of a protein is one of the states in the folding process. Which of the following options is incorrect about the molten globule state? Right? So the first option is it is a denatured state. The next one is there are hydrophobic interactions among the polar residues. Then the third one is it is less compact, and the fourth one is it is a collapse state. So yes, it is less compact compared to the native state. It is a collapse state, that is true. It undergoes some amount of folding to come to this molten globule. There are hydrophobic interactions among nonpolar residues, that is 100% true. So which is the wrong answer? It is a denatured state. That is the wrong answer because it is not denatured. It is partially folded. Right? Okay, now we go to the next question. Protein folding is spontaneous because first option is delta G is positive. Now if delta G is positive, it is not a spontaneous process. So your first option is definitely wrong. Delta S and delta H are both positive. Right? So if delta H is positive, then the delta G becomes either positive or very less negative. Then it cannot be the right answer. Delta S is negative and delta H is positive. If delta S would be negative, once again the delta G would become more positive or very less negative if delta H is positive. Correct? Based on your equation. So that also cannot be the right answer. Now delta S of water is positive and delta H is negative. Now while telling you about the thermodynamics of protein folding, I told you how the entropy of the system increases, and so delta S of water is positive, but the delta H is negative because different bond formations take place, so a lot of energy is released in the form of heat. So the right option is option number D. In protein folding, hydrophobic collapse of a polypeptide chain leads to increase in entropy of the water. Change in enthalpy is large and negative. The hydrogen bonds, electrostatic interactions, van der Waals interactions, etc. contribute to the enthalpy. Protein folding is driven by enthalpy. Right? Remember this, alright? Now, there are more questions on protein folding. Try to solve these. Which class of protein is the heat shock protein part of? First option is catalytic proteins or enzymes, chaperone-like proteins, immunoglobulins, ribonucleolytic proteins. So, we discussed about this. The right option would be chaperone-like proteins. For the second question, in the hydrophobic core of a protein, which of the following forces is responsible for hydrophobic collapse? Is it Keesum force? Is it Debye force? Is it ionic forces? Or are all the van der Waals forces involved in hydrophobic collapse of the protein? So, the answer is B, chaperone-like proteins. And for this, it is D. That is Keesum, Debye, London forces. All of these van der Waals forces are involved in hydrophobic collapse. So what is Keesum force? When there is a permanent dipole interaction, that is called Keesum interaction. What about Debye interaction? When there is a permanent dipole-induced dipole interaction. London forces are induced dipole forces. So they are always there. All of these contribute to hydrophobic collapse. They are long range forces. Okay, so remember that. Now, the interesting point is that here I have listed out many different kind of interactions which, based on the bond length, they can be seen as medium range to short range and long range forces, right? Salt bridges, they are having very strong interaction, so very small bond length. Hydrophobic interactions, on the other hand, they are having very long bond length because they are weak interactions. Okay. So now we will move on to the last topic of our session, which is studying the structure of proteins using computational tools. Now you understood about the crystallography, NMR and cryo-EM, which are very tedious and very time consuming. So how do we get to know about the structure of proteins easily just from the sequence of the proteins? So for that, different computational tools are available, and the most recent one being AlphaFold 3, which uses DeepMind and AI, right? So you can just go to the AlphaFold server, you can type and then provide a random sequence of a protein and you can check how this actually folds. Okay, so we come to the sequence structure function relationship. So based on the structure of the protein, its function can be estimated. Now based on the sequence of the protein, its structure can be found. So how do we do that? There are different ways in doing it. So you can do it by homology modeling. So homology is actually when two proteins are having very similar sequence. Then what happens? They have very similar structure. So they actually belong to the same family. They are having a common ancestor. And if one of these two proteins, their structure has been solved by, let's say, X-ray crystallography, NMR, or any other cryo-EM or any other method and deposited in the protein data bank or PDB, then computationally you can predict the structure of the other protein which is homologous to this one if the sequences are very similar and the sequences are known. Okay. Now the third one is fold recognition. So how do you find this out? You do this using BLAST. Using BLAST, you can come to know about the sequences of different proteins that are available, and then you can perform homology modeling to see if the structures are also similar or not. Okay. Now what about fold recognition or threading? So the proteins are having different domains. So these domains are functional modular units, right? So protein engineering uses these domains and fold in the protein. See, they are used to model those proteins which have the same fold as proteins of a known structure. But then they do not have homologous proteins with known structures. So let's say there are two different proteins. One of them is having a DNA binding domain and the other one is having a DNA binding domain, but it is also having a helicase domain in it, right? So then what do? Is you just make the DNA binding domain of this other protein, right? Because the DNA binding domain of this one is known, and so you go through fold recognition process and you make the domain for this protein whose only sequence is known. Then there is completely de novo modeling using ab initio modeling, which uses the laws of physics along with protein fragments to construct a model using the laws of physics, right? So this is how the protein structure is deduced from its sequence and the function is predicted and tested, right? Try to answer this question. Homology modeling algorithm is applicable for proteins belonging to the same family, proteins belonging to different families, proteins with similar amino acid sequence, proteins having same secondary structures with different linkers. Proteins must be belonging to the same family because they are homologous. Now, since they are homologous, their amino acid sequence would definitely be the same. So, 1 and 3 would definitely be there. Proteins having same secondary structures, but with different linkers. So, there are small regions of mismatch, but the rest of it is similar. So, then the right option would be 1, 3, and 4. Homology modeling is predictable, is applicable by assuming the amino acid sequence have very similar tertiary structures. Proteins belonging to the same family have same secondary structures. The linkers may vary. Okay. These are the different websites, different articles, different review papers, different books that you can refer to. Please try to refer to these and get a better understanding of this topic. It's a very interesting topic. So if you have any feedback, any questions, any kind of doubts, you can come back to me in the next class. We have the next class on the next Tuesday from 6 p.m. to 8 p.m. You're all welcome to join. Okay, thank you for your time. Thank you for watching the video. We'll meet again in the next class.